So I'd like to start my talk. My talk is about the substrate-borne vibrations reduce the infestation of the tobacco whitefly, Bemisia tabaci, experimental assessment in tomatoes from lycopersicum. So actually, I am the collaborator with Sakuma, and we are doing some applied research on the management of whiteflies using tomatoes. So I'd like to start. Sorry. Yeah. So first, I'd like to introduce the tobacco whitefly, Bemisia tabaci. Probably, I think many people knows what is the tobacco whitefly, but it's a very serious pest, and this species is known as a global significant agricultural pest that causes serious damage to vegetable, flower, and grain production by sucking pollen. So, and this species transmit many kinds of virus, such as begon virus, green virus, epom virus, and so on. And in Japan, we are suffering much about the tomato yellow leaf curl virus, which is called TYLCP. So, it's also known that this species contain more than 40 cryptic species. These are called as biotypes, and especially the B and the Q biotype are most invasive, and these are so problematic also in Japan. And what is more about the problems also of this species? So the resistance against pesticides, many kinds of pesticides, is also problematic. So without relying only on pesticides, we cannot do much more for anything. So that's why there is a demand for combining other methods for controlling pest effectively. So we know that many insects can be influenced by the perception of visual, chemical, and tactile stimuli, but also of acoustic and vibrational cues. So we can raise some examples for that. And the first is the behavior of startle response. And this is a study of Takuma and his colleagues. And I can show you this example. This is a species of Japanese pine sol beetle, Monochamus alternatus. And the Takuma and colleagues have clarified that there is some particular sensory neurons inside of the legs, and this is called scoparium, and another structure it's called cuticular apodeme. So these consist of the femoral called notonatal organs. Sorry, it's called FCO, and this scoparium is consists of many fibers of neurons, such as this. And if we destroy these organs artificially, this species cannot respond to external stimuli, and they lost the startle response. So that's very interesting phenomenon, I think. And the another example is egg hatching, and this is related to the fitness of this species. And this is a work of Hiromi Mukai. She talked yesterday. And this is a species of the pentatomid bug. The species name is Parastrachia japonesis. So in ordinary situation, the females protect her eggs just like this. And this is female, and she's protecting her eggs like this. And actually, the female emit some signals to the eggs, so-called vibration. So she vibrates a little bit onto this the egg ball. So the nymphs start to hatch simultaneously, and they start to move, and they move to slightly a different position, and then to start to suck some kind of nutrients. And without a female, the hatching is also start, but the hatching will be delayed without females, and then the hatching is usually continues for about 1 day, but some of them cannot hatch normally. And actually, the whiteflies, it's also known that whiteflies duet with vibration as a mating signals. So this is some examples using the different biotypes, which was conducted by Kamiya, Dr. Kamiya. And the first one is the mating call between biotype Q male and biotype Q female. And they, so the first, the male emit the signal, and the female respond against it. And this calling, continuously repeated, just like this. But if this biotype B meets the biotype Q female, first they emit some signals, but after several seconds, the females doesn't respond anymore, and males emit his signals. So this is very interesting sound vibration communications in whiteflies. So that's why we consider that the applying vibration is expected to affect the behaviors of tobacco whiteflies. So that's why the signals may disrupt the mating behaviors and ovipositional behavior and feeding behaviors. So this is experimental setting. So these are the pictures which we conducted in the field, and we built this kind of house, and then we inserted these plants inside. And this is the schematic diagram of the experiment. And the vibration parameters. So we applied the 1 second pulse with 100 Hz and 9-second intervals was repeated. And the exciter was driven for 1 minute per 10 minutes during 7 in the morning and 6 in the evening during the daytime. And we measured the intensity of vibrations, and the intensity is between the intensity was between 7 from 7 to 76 meter per second squared. And I think these intensities are enough for particular distance from the exciters. So, but later I'll talk about attenuation of these signals. And this is measured, and we used the 30 individuals of 1 to 9-day-old adults, and this was installed on each tomato stalk. And this is the picture. This is the figure of this experiment, and here is a vibration exciter, and we set the tomato plants like this, and we also set the sticky trap between the tomato plants. And this is a picture of a vibration exciter, which is made using giant magnet-stricted materials. So, this was attached on the pole, plastic poles, and then it's connected with a plastic bar like this. And the vibration starts to emit by this plastic rod. And in this study, we focused on the three matters. The first is the evaluation for pest control. So we counted the number of whitefly, larvae, and adults on any 3 compounds leaves of each plant. And second is to evaluate for plants. So we also counted the number of flowers since the start of the experiment. And the third is the evaluation of fruit quality. So, we compared the characteristics of harvested fruits. So that, for example, the weight and height, weight, and the sugar and the acidity. So I'd like to start to explain the result. And the first one is temperature changes in the number of nymphs. And these graphs consist of the different the consequence of the temporal change in the density of whiteflies in each house. So in both houses, that's why the house 1 and 2 compared to the control. Control means without vibration treatment. So so the population density is clearly decreased in the treatment of vibration compared to the control in both houses. And finally, in average, 46% reduction was obtained. And the previous result is for nymphs, and the next is for adults. So the tendency is very similar, but the density of the temporal change is so fluctuating compared to the result of nymphs. But finally, we got 28 reduction in average. So this is the temporal change in the number of flowers. But in this case, we did not find any significant change in the number of flowers. And this is about the number of trapped whiteflies. So, so we did not find any significant difference between control and vibration in the number of trapped whiteflies. And this is the effect on fruit characteristics. So we measured 5 different fruit weight, fruit characteristics such as the fruit weight and width and height and the fruit sugar content and fruit acidity. So overall, we did not find any significant difference in these kind of characteristics. But the fruit acidity is a bit inflated when the vibration is adopted. But this is also not significant. So in conclusion, we can control the whitefly density by using the vibrations, and we also find there's no adverse effect on plant growth and fruit. So in the future, so we are planning to consider how to manage the pollination by using vibrations. So in Japan, so it's prohibited to use the bumblebees because this is the invasive species. So that's why we are studying this aspect of the vibrations. And so what is more? So we are seeking the proximate factors that cause reduction of whiteflies. So as Valeria and other colleagues studies the behaviors of the whiteflies, so we will have to study the similar things such as disturbance in mating signals and reduction of position and delay in developmental stages. So that's it. So I'd like to express my appreciation to all members of the research group and other staffs. And thank you very much for so thank you for my attention. That's it.
Thank you, Haruki. Your talk